Okay, take two. I did just record this, but let's get into it. So this is going to be a genetics class, a kind of lecture series for year two biology students at university, going over some kind of advanced genetic stuff. This lecture is going to be focusing on DNA packaging and the epigenetic consequences of it. So, let's get into the basic ideas. DNA can be packaged very efficiently into cells, which do not have a very large area, but we have a lot of DNA in our cells. So how do they fit all that DNA within such a small little area? Well, they wrap the free-floating DNA, called chromatin, around these structures called nucleosomes. But when the DNA is wrapped around the nucleosome, it can't be transcribed. It can only be transcribed when it's then unwound from the nucleosome. We discovered nucleosomes by cleaving DNA with a couple of nuclease enzymes called DNase 1 and micrococcal nuclease. We ran a gel from the cleaved DNA, and it was found to have been cleaved at regular 200 base pair intervals. So if we want to take a look at this image, this would be our free-floating chromatin, which we couldn't fit into the cell. So we wrap it around these blue things called nucleosomes, so it takes up less space. And then we did an experiment to discover that these things existed by taking this DNA here, which is what it looks like in our cells, treating it with a couple of enzymes, DNase 1 and micrococcal nuclease, which would cut the DNA at points where it could cut the DNA, which would be in these regions here that are not wrapped around the nucleosomes, and it was found that it cut them at intervals of 200 base pairs. So you can think, we've got here 200 base pairs around here, cut it. 200 base pairs in between the last cut and this cut it. Then cut it again. There are two levels of packaging of this free-floating DNA into this nucleosome associated DNA. The first level of packaging involves the DNA, the chromatin, wrapping around these nucleosomes, which are made of 8 histone subunits, and they are held in place by a structure called a linker histone, which prevents the coiled DNA coiled around the nucleosome from detaching. So it's not shown in this picture, but see these little black things in the middle of these kind of flowery structures? They are the linker histones preventing the chromatin from unwinding from the nucleosomes. Then we have the second level of packaging that has the nucleosomes condensing into an alpha helix, which we can see at the bottom here. So an alpha helix is kind of like a turning structure of all these little flowery things. And each of these little flowery things has 6 nucleosomes with the chromatin wrapped around them, and 6 linker histones preventing the uncoiling. And each one of these kind of like 6 nucleosome structures we call a solenoid. So that's how we package DNA very efficiently within cells. Gene transcription of this free DNA relies on controlling proteins, recognizing promoter sequences, and binding to them. But like I said before, they can only bind to promoter sequences when the DNA is not wrapped around the nucleosome. So these proteins can only recognize activated sequences in the sections of free DNA. So it has to be unwound in order to be transcribed. Activator proteins bind to sites of DNA called enhancers, and they speed up the rate of transcription. Co-activators are adapter molecules that work in tandem with these activator proteins that relay signals from the activators to these things called basal transcription factors. And these transcription factors essentially are responsible for putting the RNA polymerase, which is the thing that is going to transcribe a gene to produce a gene product that we want for pre-mRNA, and then that gets spliced to mRNA, and then that gets translated at a ribosome to produce our polypeptide, which then gets folded in the Golgi to produce our functional proteins. So if we think about this free DNA as its own kind of gene, so imagine this is just a section of the wider genome, we have our activator protein recognizing an enhancer that would be somewhere around here. Our activator protein recognizes it. Co-activators are working in tandem to the activator protein to relay signals from it to these basal transcription factors, which then bring RNA polymerase here, and obviously you need your helicase to unwind the double-stranded DNA for the transcription to occur, but we're not going deeply into transcription here, just about the idea. So the RNA polymerase works here, and then it would follow it along, and it would transcribe a complementary strand of pre-mRNA that would go through the other processes I described before. On the other side of that, we have repressor proteins, which bind to sets of genes called silencers, and they interfere with the functionality of activators, slowing down the rate of transcription. So the repressor proteins are just preventing the stuff that the activator proteins are doing, so you're not getting your RNA polymerase at the start of the coding region of a gene, so the gene isn't being transcribed. In order to take our DNA from this condensed form, where it's associated with the nucleosomes and linker histones, we need extracellular signals to tell our cells that it's time to unwind the DNA in order for transcription to take place. We found that out in this experiment where chicken erythroblasts, which are stem cells found in the bone marrow of chicken bones, which are precursors to cells called erythrocytes, which if you do any kind of medical modules, you'll know are just the fancy word for red blood cells. So these erythroblasts, red blood cell stem cells, typically synthesize globin at 14 days within the chicken. An experiment was done where the chicken erythroblasts were taken from the chicken and cultured independently from the animal, and these cultured cells were treated with DNase-1 and BAMH1 nuclease enzymes. And it was found that the cultured cells were both not producing globin and they had not been cleaved by the enzymes, whereas a control experiment that took erythroblasts from the living animal, they were producing globin and they had been cleaved. So why do you think they hadn't been cleaved by these nuclease enzymes? Well, because in order to be cleaved and to produce the globin to be transcriptionally active, the DNA would have to be in its free DNA state, but because there were no extracellular signals telling the cell to unwind its DNA from this state, it was stuck in this state, so the chromatin in the DNA and the genes that it codes for the globin gene was still wrapped around the nucleosome, so it could not be cleaved by the enzymes and it could not transcribe the globin protein.